Okay, this chapter we're going to be talking about the cell membrane, the skin that separates from the inside of a cell from the outside of a cell. Okay, we talked about before, last unit, we talked about how phospholipids make up the majority of what a cell membrane consists of. Okay, so the membrane is like a wall around the city, but it's not just a wall, it's spherical because cells are spherical. So it's a barrier separating the outside environment from inside of the cell. Okay, so we said before that cell membrane is composed primarily of phospholipids. Phospholipids are molecules that are amphiphilic. They have a hydrophilic head, phosphate group, and then they have a hydrophobic tails, two of them. Those are fatty acids or hydrocarbons, tails. Okay, the way the phospholipids are orientated is as so in the second picture. It's a phospholipid bilayer with two layers of phospholipids. Notice how the fatty acid tails point towards each other with the polar heads on either side of the cell membrane. The intracellular side is the side that's touching the cytoplasm, and the extracellular side, so it's called the interstitial fluid, or the fluid that bathes the cell, the external fluid. So the inside of the cell and the outside of the cell is a watery solution. Aqueous means a water solution where the solvent is water. Okay, so that's why the phospholipids are arranged with the polar head, the hydrophilic head, pointing towards aqueous solutions, either outside solution or the intracellular solution. And the hydrophobic fatty acid tails kind of waterproof the cell from losing its contents or from letting anything just come into the cell. Okay, a cell has to maintain a stable internal environment different from the environment outside the cell. Okay, this is homeostasis on a very basic level. Homeostasis is regulating a constant, somewhat constant situation. So, membranes are said to be selectively permeable or semi-permeable. That's another name. I'll usually use that because it's easier to say. So only certain things are allowed through the cell membrane. So you got these yellow molecules and these green square molecules, and it looks like the cell membrane is semi-permeable to the green molecules, but it is not permeable at all to the yellow molecules. Again, I'm pointing out that the membrane has a very important purpose, function, in regulating the amount of nutrients and waste products crossing the cell membrane. And again, that's an example of homeostasis on a very basic level. Okay, so cell membrane, those phospholipids are not just glued together. They're wiggling around. Molecules move around. They have motional energy whenever they're at any temperature above absolute zero. The warmer they are, the more they bump around. So as they shift around, certain molecules can get through. Certain small molecules, water, for example, small nonpolar molecules can, such as oxygen and carbon dioxide. Okay, so just with the phospholipids alone, water and nonpolar molecules such as oxygen gas and carbon dioxide can get through. The gaps that form when the phospholipids are moving. Now, it's not just phospholipids at the cell membrane, or also called the plasma membrane. It's not just phospholipids that are in the cell membrane. There's also proteins that are like gates. So I'm going to show a protein here with a hole, a pore in it, like a toilet paper roll. That's embedded in the cell membrane in between the phospholipids. And it allows other molecules to get through, larger molecules, polar molecules that normally would not, bigger polar molecules that normally would not get through because there's no water in between the polar heads of the cell membrane, and that would repel the bigger polar molecules. Water molecules are polar, but they get through because they're so small compared to larger polar molecules like glucose, amino acids. Also, charged ions can get through with the help of proteins, because charged ions are always surrounded by water molecules, making a big shell of water molecules around the ion, making kind of a big thing that won't get through the nonpolar fatty acid tails unless it's going through a protein gateway. Okay, I already taught you this. All solutions in biology, in living things, are aqueous solutions where the solvent is water. Now I'm going to talk about how things get through the cell membrane more specifically. Now there's two ways a cell membrane can let things through. Passive transport mechanisms. No energy is needed to run these gates that let things through. The particles that are moving through the cell membrane just move through spontaneously with no energy input. Active transport requires input of energy. Quick review. The section labeled A. Okay, those are fatty acid tails of the two layers of phospholipids. Okay, fatty acid tails are hydrophobic, nonpolar parts of the molecule. Now, if they said if that was letter B, section labeled B would be amphiphilic. It would have a polar end where the red ball is and nonpolar tails. This question is pointing out the red ball, which is the phospholipid head, the polar head of the phospholipid, the phosphate head. And so that part would be hydrophilic. And you have it over here too, of course. So, sorry, I pushed the button already. How many layers of phospholipids? Okay, we've seen many pictures so far of the phospholipid bilayer, and it's called the bilayer. So there's two layers. This question is kind of hard because you got to visualize what they're talking about with each of these choices. So phospholipids bilayer, how do they arrange themselves? Well, let's draw a picture that you know already. I'll just draw one phospholipid in each of the layers of the phospholipid bilayer membrane. Do the hydrophilic, okay, that would be the polar heads. Do they orientate pointing towards each other? No. They're pointing away from each other. Do the hydrophobic parts contact each other? Yeah, look. Hydrophobic parts are the fatty acid tails. Those are in contact with the other phospholipids on the other side. And they're in contact with each other as well in each layer. So that's the one. The hydrophilic ends of one layer meets the hydrophobic ends of the other layer. That would be like this. That doesn't happen. The hydrophobic ends contact and close. So that'd be like this. And that doesn't happen. Plasma membranes allow all types of molecules to pass through. No, they're semi-permeable. They're not going to let everything get through. Okay, so regulation of the internal environment of a cell or of an organism, you should know by now, is called homeostasis. Okay, which is true about phospholipids. They are completely insoluble in water. No. Recall that phospholipids have a polar head and nonpolar tails, so they're amphiphilic. They have parts that are polar and parts that are nonpolar. So they're philic for both types, like an amphibian likes the land and the water, both. So they are not completely insoluble in water. They form a single sheet in water. No, they form a phospholipid bilayer. They form a structure in which hydrophobic portions face inward. They form a selectively permeable membrane. That's it. Letter D. Okay, let's talk about passive transport. You recall that passive transport means no energy needed to run it, to transport things across the membrane. Again, passive transport does not require any energy. It just happens spontaneously. Okay, it happens whenever, let's say this is a cell membrane. Whenever there's a larger concentration of a solute, let's say this is a solute, dissolved in water, of course, but that represents a solute. And let's say this side has fewer solute particles, this side of this cell membrane. Okay, well, if the molecules can move through the cell membrane, either through the phospholipids or protein gates, then what will happen is those particles will move from a high solute concentration to the side with low solute concentration, and that will happen without the need of energy pumping it. It just happens. Actually, it gives off energy, and you can use that energy to do work, but we'll find that out later. It does not input energy. Okay, there's 3 types of PTs, passive transport. Diffusion, I call that simple diffusion. Osmosis, and then facilitated diffusion. Let's talk about each of these types. Okay, so diffusion is the random movement of particles from a high concentration, from a high solute concentration to a low solute concentration. Okay, so it's any particles. So it's very general. This picture is showing you put a drop of blue food coloring into a glass of water, and that drop will diffuse throughout the water from the drop where it's highly the dye particles are highly concentrated to the rest of the water where dye particles aren't highly concentrated. If you open a bottle of perfume or someone passes gas, pretty soon the people next to that person will smell it. Then further on, those guys will start smelling it as the perfume particles diffuse throughout the room. Again, this is passive transport, simple diffusion. No energy is required for this to occur. Okay, so I just told you the definition of diffusion is when particles move from a high concentration, a high solute concentration, to a low solute concentration. So look at this picture. It looks like outside the cell, we got a whole bunch, a high concentration of those little blue hexagons. Eventually, they'll diffuse through the cell membrane, maybe through a protein gate, maybe just through the phospholipid layer, and until both sides have the same concentration, if that's possible. And then diffusion net diffusion will cease. Now, what do I mean by that? Well, since diffusion is a random process, anytime, at any point, we have the solute moving in both directions. Well, not at the very beginning, it only can move through the membrane. particle can only move through the membrane. But once it's on the other side, some can move back through. Some can just move right back through. But there is a net motion. There's a net movement, a bigger flow inward than outward. So that's why we say net movement. Even when they're at the same solute concentration, diffusion is still occurring at a constant rate, back and forth. We call that equilibrium. The concentrations are the same on both sides of the membrane, but that doesn't mean, that means net diffusion, net movement of particles. That means total overall motion of particles is zero. Some are moving into the cell, some are moving out at an equal rate. Look at this picture here. The green dots represent oxygen molecules. Okay, so oxygen and carbon dioxide can diffuse freely, simple diffusion, through the cell membrane. So I see, if I take a little area here and I compare it to the little area inside the cell, well, we have a higher solute concentration, solute being the green balls, being the oxygen gas, higher solute concentration outside than inside. You see lesser green balls in the same area square. Okay, so there'll be a net flow. There'll be a net flow by diffusion inward into the cell. But that doesn't mean some aren't going back in. The net flow inward means that the rate of molecules moving inside the cell, into the cell, is greater than the rate of molecules moving outside the cell. So for gas diffusion, whether it's carbon dioxide or oxygen, the transport happens spontaneously. No work needs to be done in order to have oxygen or carbon dioxide diffuse down a concentration gradient. Okay, I just used the term down a concentration gradient. Okay, a gradient is a hill. If you're going from a high solute to a low solute concentration, like going from a lot of dissolved solute to a place where there's less dissolved solute. Diffusion happens down or along a concentration gradient. So diffusion direction, net diffusion direction, down or along the concentration hill. We say gradient. So you're going from high to low. That's moving across. That's moving along or down the concentration gradient. Gradient means hill. Now, that requires no energy is required. No energy input is required. Later on, we'll find out that you could get energy from this happening, because just think like water flowing downhill. We can we can take the energy out of that water that it had uphill with like a paddle wheel. So like water, you put a paddle wheel here, and this will cause the paddle wheel to rotate, and the paddle wheel might have electrical generator on it, and then you could sell that electricity from the motion of the paddle wheel that was originally from the motion of the water molecules to people. Here they're using the term concentration gradients of different solutes. Okay, so they're independent of each other. For example, I don't know, if you had, let's say, a semi-permeable membrane here, and you had these guys right here, and then let's say you had these solute particles. Okay, so there's a higher concentration of the dark circle ones, the balls, on the left side. So it'll be a net flow of those particles through the let's say that's a cell membrane. The net flow of those guys will be down the concentration gradient, or along, as they say, along. From where there's more balls to where there's less. Okay, but the triangles will have a net flow this way. So net flow of the triangles is also down the concentration gradient, or along the concentration gradient, but it's moving the opposite direction because there's a higher concentration of triangles on the right side compared to the left side. So net flow or triangles will be from the right to the left, down its concentration gradient from high to low. Okay, so we've been talking about simple diffusion for very small molecules. For larger molecules, you can have diffusion, but you need a special protein gate in the phospholipid bilayer that will selectively allow these bigger molecules, or these charged molecules, atoms or molecules, have a partially charged molecules. And larger molecules, they don't do simple diffusion, but they do facilitated diffusion. And facilitated diffusion means you need a transport protein. Now, no energy is needed still, because this is an example of passive transport. Okay, an integral protein is a protein that's embedded in the cell membrane. An example would be these gateway proteins that allow certain elements or particles to get through the cell membrane. Now, water does get through the cell membrane like carbon dioxide by simple diffusion. But, and oxygen too. But, I don't know why they had this arrow right here. Okay, but when it goes from a high concentration to a low concentration, that's simple diffusion. So the flow should be this way. Now, water does get through, but it has kind of a tough time because it is polar. So it gets hung up. It slows down when it gets to the hydrophobic tails. So the cell has to make it quicker, diffusion of water quicker, called aquaporins. Think of water with a water pore. And it facilitates the water moving down its concentration gradient and makes it happen quicker. So even though water can do simple diffusion, it more efficiently diffuses through protein with a pore in it that allows it to travel through. Okay, this is called facilitated. Facilitated diffusion means diffusion that's helped by a protein. Okay, so there are two types of integral proteins that can do passive transport. Aquaporins are an example of the channel proteins. In the channel proteins, there's no binding of the solute to the protein. It just passes through this channel, this hole, this pore. Okay, so there's no binding. There's no chemically bonding or attracting the solute to the protein. And they only go down a concentration gradient from high concentration to low concentration. Okay, carrier proteins are similar. They're integral proteins embedded in the cell membrane, but they bind to a specific solute and causes the carrier protein to change its shape in such a way that it can then eject the solute particle. Now, weird thing, carrier proteins can flip around and be active transport. When they're doing active transport, they need energy, of course, and they can push the solute, pump it against the concentration gradient. So passive transport, the solute moves along or down a concentration gradient of the solute. Both carrier proteins and channel proteins can do that. Active transport, the solute moves against or up a concentration gradient this requires energy. You gotta pump it. You gotta put energy in it to it. Doesn't happen like in passive transport. So I said carrier proteins can either do passive or active. Okay, let's see. Review. Diffusion is the random movement of molecules in all directions. Okay? The net movement of diffusing molecules is not against as active transport along their concentration gradient. All molecules diffuse inside of membranes. No, that's false. Depends on the concentration gradient. Each type of molecule diffuses at a rate that is independent. Yeah, because you have different concentration gradients for different solutes. So it doesn't depend. One type has its own gradient and behaves differently than another type of solute with a different concentration gradient across the membrane. So this is true. Okay, so the bag is like a cell membrane. You can call that a cell membrane. It's permeable. Okay. It's permeable to water, glucose, and fructose. Those are two monosaccharides. The concentration of sucrose outside the bag is greater. So I'm going to say bigger sucrose concentration. My symbol for concentration are square brackets. This is smaller sucrose inside the bag. Okay, the concentrated fructose is smaller than inside. So fru is smaller than the fru concentration inside. Which side will exhibit a net diffusion into the bag? Well, the one that's going against the concentration gradient from high to low. So it wouldn't be fructose. It would be from, if you into the bag, it would be going from the one that has higher concentration outside than inside. So that'd be sucrose. When diffusion has occurred until there's no longer concentration gradient, then equilibrium has been reached. Well, equilibrium doesn't mean that there's no more diffusion happening. Just the diffusion of those solute particles is equal in both directions. Okay, facilitated diffusion is passive transport, no energy input. You don't have to plug it in. And it always goes down a concentration gradient. Okay, with that knowledge in my head, I'm going to read these questions and figure out which one's a good one. So the passive transport moves molecules against their, no, it moves them with their concentration. Again, wrong. Along is the same as with, but it doesn't use energy input with their energy, without the use of energy, so it's letter D. Carrier proteins are examples of integral proteins. An integral protein is something that's embedded in the membrane, so that's true. What you want to find is not involved with facilitated diffusion. No, you got a concentration gradient. You got a cell membrane. Facilitated diffusion is through a protein in the cell membrane. So you have to have a cell membrane. You need a protein, but you don't need an energy source because there's passive transport. So that's the one that you don't need. The third type of passive transport mechanism that I've left to the end is osmosis. Osmosis is net diffusion of water molecules through a semi-permeable membrane into higher solute particles, or you can think of into a place where there's less water. That's what diffusion is, moving from a high concentration of solute to a low concentration of solute. That's if you're a solute particle. But if you're water, you follow the rule. You're going from a place where there's more water to a place where there's less water. Or you can flip it around and say, water moves from a place where there's low solute concentration to a place where there's high solute concentration. So let's do a few examples here. What if you had a bag made of a semi-permeable membrane that was in a solution of, let's say, 5% of the molecules were salt ions. So 5% concentration salt. So that'd be 5 grams of salt for 95 grams of water. And then inside here, you had a 1% salt solution. Now, the membrane, the material of the bag, will only let water through. It won't let salt ions through. Okay, so it's only permeable to water. Okay, so predict what will happen to the bag. Well, will water enter the bag or will water leave the bag? Because salt can't enter or leave because the membrane is only permeable to water. Okay, so you tell me, water always goes to the saltier or the more concentrated solution. So what will happen, there'll be a net flow of water out of the bag because 5% is more concentrated in solute than is 1%. So there'll be a net flow of water out. Of course, there'll be some water that flows back in because this is diffusion. Diffusion works both ways, but there's a net flow out of the bag. That means there's more water flowing out than is flowing back in. So what will happen in the bag? The bag will shrink in volume as water leaves. Okay. And if there was a bag in there, And it was, let's say, 20% sugar solution. And outside the bag in this solution, there is, let's say, 5% sugar solution. It doesn't have to be sugar, it could be anything. I could say salt, because I'm going to tell you in a minute that the membrane is, again, semi-permeable only to water. Just like in the previous case. So it's the number of dissolved particles, not the identity of the dissolved particle, the solute, sugar, or salt. Okay, so water will net flow in what direction? Always to the higher solute concentration. So there'll be a net flow of water into the bag. The bag will increase in volume as water flows into it. Again, there's some flow out, more flow in. So there's a net flow into the bag. If you rather think of it this way, if a solution is saltier or higher concentration, solute concentration, there's less water, and water will move from a place where there's more water, a lower concentration, to a place where there's higher amounts of water, a higher solute concentration. So the diffusion of water through a semi-permeable membrane is osmosis. Now it can go through the lipid layer, the phospholipid bilayer, between the phospholipids, or it can go through a channel protein, a water channel protein called aquaporin. In either case, it's called osmosis. It's the diffusion of water through a semi-permeable membrane. So osmosis is simple diffusion, or could be like here. This is a channel protein. That'd be facilitative diffusion. But in either case is osmosis. Whether there's a protein channel protein, or whether it's just going through the phospholipids without a protein. Okay, tonicity, the tonicity of a solution. It is a characteristic of the solution outside the cell. Okay, so if you have a hyper means like hyperactive, over, a lot. Hypo means low. Iso means equal. So hypertonic means that surrounding the extracellular solution has a higher solute concentration than inside the cell, the intracellular solution. Hypotonic means the outside solution has a smaller solute concentration than the inside cell. And isotonic means the solute concentration is equal inside and as it is outside the cell. Okay, in this picture, the green things are solute molecules, and the black things represent water molecules. Okay, let's look at what kind of situation is this cell sitting in? What kind of solution? Is it hypertonic, hypotonic, or isotonic to the inside of the cell? Well, I make a little window in my mind and the same size, and I do one inside and one outside the cell. And I look, well, outside the cell, I have more. In one of these windows, I have more. Looks like I got 3 right here. I got 3 solute molecules, and I got like half of one. So there's more solute. There's a higher solute concentration outside the cell than there is inside the cell. So let me write that here. A higher solute concentration outside the cell than inside the cell. Therefore, it's hypertonic. If you have more solute outside, it's called hypertonic to the inside. So therefore, there'll be a net flow of water to the higher solute concentration. That would be, in this case, out of the cell, because the solute concentration is bigger outside the cell than inside the cell. But there will be some flow of water back into the cell, but much more out than going in. So there'll be a net flow out. I should have mentioned that the cell membrane is permeable to water, but not to solute. I should have said that, or else the solute would have obviously had a net flow inward. In this net flow of water down, up the solute concentration gradient is called osmosis. Osmosis is when water moves against a solute concentration gradient, if you want to look at it that way. It moves from a place where there's higher concentration of solute to a place where there's lower concentration of solute. And recall that the place where there's a lower concentration of solute, there's more water concentration, if you want to think of it that way. So water is flowing. Water is flowing from a high water concentration to a low water concentration. One of the videos I gave you guys talks about water potential. And water potential is almost like water concentration, solvent concentration. So if you have a low concentration, that's kind of a weird way of looking at it, but it might be helpful. If you have a low solute concentration, you have lots of water compared to solute. Means you have a high water concentration or a high water potential. If you have a high solute concentration, that solution has a low water potential. And osmosis is basically the net flow of water from a high to a low water potential. Or the net flow of water against the solute, the sugar or salt dissolved, the solute concentration gradient. because I could rewrite this as the net flow of water from a low solvent, solute concentration, sugar or salt, to a high solute concentration. So for this one, again, hypertonic means a high salt concentration outside. High solute, not salt necessarily. High solute concentration outside the cell. Then inside. So osmosis has the water going from the low salt concentration to the high salt concentration, or from the high water potential to a lower water potential. So what's going to happen to the cell? When the water leaves it, it's going to shrink or shrivel up. Okay, here we have a cell in a hypotonic solution. The solution that the cell in is hypotonic relative to the inside of the cell. Okay, so make your little window. You can see 3 solute molecules. Do the same thing outside the cell. And you can see 1 or none. Okay, so the hypotonic means a higher, hypo means lower. So lower solute concentration outside the cell than inside. Or, if you want to do that, a higher water potential outside the cell. Outside the cell than inside. Okay, so water potential and solute concentration are opposites. They're inversely proportional. So what happens is the water is going to flow from the lower solute side to the higher solute side. And the net flow will be that. Again, this is osmosis. What do you think is going to happen to that cell? As the water flows in, the cell is going to swell. And if it doesn't have a cell wall, it will burst. Okay, so plant cells protected from this with a cell wall, but animal cells aren't. So you put a red blood cell in distilled water, and it's going to blow up. Okay, so in an isotonic solution, supposedly the solute particles are supposed to be just as concentrated inside as outside. This picture is poor. Let me try to fix it. So now the concentration of the green balls looks about the same outside and inside the cell. So we say the solution is isotonic to the solution inside the cell. The outside solution is isotonic to the inside solution inside the cell. Okay, the water flows equally. Water will osmose equally in both directions, into and out of the cell at the same rate. So there is no net flow because they cancel each other out. I mean, there is flow in, but the flow in equals the flow out. Okay, so easy way to remember: hypertonic, hypotonic, isotonic. Hyperactive means more. Hypothermia means low, less. Iso means the same, like isosceles triangle. Okay, so Hypertonic means solute concentration. Okay, so that should be an easy way to remember the three types of solutions outside the cell compared to inside the cell. Okay, so solution A is usually the outside cell solution, and solution B that you're comparing to is inside the cell solution. So this table kind of puts it in perspective. Solute concentration. Okay, we're talking about the outside solution in all cases when I ask you questions. Compared to inside the cell solution, where's the greater solute concentration? If a cell is in a hypertonic solution, the solution is greater outside the cell than inside. Where's the water potential? Okay, so when they talk about water concentration, we call that the water potential. So that's where the water is more concentrated, and where the solute is less concentrated. So hypertonic solutions, therefore, outside the cell have a lower water potential because it has a higher solute concentration. So inside the cell is where there's less concentration of solute. You would have a higher water potential. Where's in that direction of water diffusion or osmosis? Well, water will go from a high water potential to a lower water potential. So since the inside of the cell has a higher water potential in a hypertonic solution, it will flow out of the cell. How would you diagram it? Well, you have more water leaving than entering the cell, and the cell will shrink. When it's in a hypertonic solution. Okay, hypotonic solution is where you got more solute inside than outside. So flip it around. Water potential is higher outside the cell, and so water will force itself by osmosis into the cell, and the cell will swell, maybe burst. Isotonic, you have the same concentration of solute, so inside and outside. You have the same water potential inside and outside. The water will flow equally inside as it osmosis outward, so there will be no net flow, and the cell would retain its original shape. Okay, so these should be easy. The diffusion of water molecules across a selectively permeable membrane is called osmosis. Only water is involved with osmosis. So water molecules osmose or diffuse from high water potential to low water potential, which means low solute concentration to high solute concentration. So inside plasma membrane, outside only? No, it depends on what side is hypertonic. Outside the plasma, no, it depends. From areas of high solute concentration No. It goes from areas of low to high. From areas of low solute concentration to areas of high. Okay, that's it. Or like I said, high water potential to low water potential. Diffusion osmosis, no. They're passive transport. No energy is needed. So false. What types of environment has a higher concentration of solutes? Hyper, higher, tonic. What type of environment has equal amounts of solute inside and outside of the plasma membrane, the cell membrane? That'd be iso, equal. What type of solution outside would result in a greater flow of water to the inside? Okay, so H2O would flow in if this was saltier and this was less salty, or solute. I just say salt, it could be sugar. Okay, so what kind of solution is this? Well, outside it has a lower solute concentration outside the cell. So that would be, by definition, hypo, lower, tonic for solute, hypotonic. Okay, so this problem you got to read really carefully. So inside the beaker, the concentration of glucose and fructose is greater. So I'm going to say high glucose, low glucose inside. Let's do inside as well. So this is outside. This is inside the baggy, deep, semi-permeable membrane. Baggy. Okay, so this has low glucose inside the bag. So it also has more fructose in the beaker, so this would be lower fructose. And in the beaker, you have a smaller amount of sucrose. And you got more sucrose inside the thing. Before osmosis begins, is the solution in the beaker isotonic, hypotonic, or hypertonic? Okay, so it depends. It's a dumb question because they need to give us actual values. How much more glucose, and so you can't really tell. So none of these really are right because they didn't give me numbers. Now, if they had given me numbers like this, Okay, so glucose, let's say the solution is 5% glucose, 5% fructose, and 25% sucrose. And this glucose was 20% glucose concentration. And let's say 10% fructose concentration. And the sucrose was what, less? Yeah, so let's say that's 5% sucrose. Well, you got to see, you have to add those up. So see who has a total bigger concentration. So 20 plus 10, that's 35% solute concentration. And outside, but inside, in my example, I have 35, it happens to be 35, 25 plus 10. Also inside, I have 35%. Now, these numbers, depending what these numbers are, depends on your answer. This was too vague to determine. I would say, I don't need help. I need more information to be sure. You can't answer for sure. Now, if they give me the percentages that I just gave you for the solute concentrations, then you would say, oh, it's isotonic. But all they had to do is change one of these numbers, and it wouldn't match that answer. Okay, which is most likely to diffuse freely through the cell membrane without involvement of a transport, without needing a channel protein. Okay, you should know these. Gases like carbon dioxide, oxygen, ions don't. No. Glucose is too big. Polar molecule. No. DNA, no way. It's way too big. So of these choices, carbon dioxide, oxygen, water would even be a good example. Answer. So small gases and small molecules of gases and water, but not ions or bigger molecules. Which of the following process includes all the others? Well, osmosis is diffusion of water, diffusion of a solute across. Well, water isn't a solute. Facilitated diffusion, passive Okay, so passive transport. These are examples of passive transport. So letter D includes the other ones. Okay, active transport as opposed to passive transport requires energy. Okay, and also requires membrane proteins that are powered by energy. Okay, so passive transport is the movement of solutes from a high solute concentration to a low solute concentration. That's passive transport. That's if solutes are moving. Now, if it's water, like we said before, it goes from a high water potential. If water is the thing being passively transported, again, I'm just reviewing passive transport right now. It goes from a high water potential, which would be a low solute concentration, to a low water potential, which would be a high solute concentration. But in either of these cases, this passive transport, you don't need any energy to drive it, to pump it that direction. Active transport requires energy, and it moves the solute against a concentration gradient. So it moves it from or down. Up, if you want to, up instead of down, a concentration gradient. Okay, so that requires energy. Think of a pump. You have to put air in a tire. You got to use energy to put more air in the tire than is outside the tire. The pressure on the tire is higher than the pressure of the air outside of it. So that requires energy. So whenever you have to move solute against the concentration gradient or up a concentration gradient, this requires active transport. So, I'm just repeating what I just said here. Active transport uses membrane proteins to move solutes through a transport protein against or up the solute concentration gradient. Okay, so it takes energy to paddle a boat against the current or up the current. Okay, the source of energy is ATP, which we'll talk about in a few in other units too. ATP is the energy coin that can be spent anywhere you need energy. So this little star represents ATP. ATP is adenine triphosphate. It will break off one of the phosphate groups of the 3 triphosphate, and that will release a lot of energy. Very important process that all animal cells use, but it actually is how nerves transmit their nerve impulses to muscles and other nerves. Is this active transport mechanism is the sodium-potassium pump. Now look at the green things in this picture. On this side you have a high concentration of sodium. On the outside of the cell, on the inside, not so great. Okay, so you also have a low, K is the symbol for potassium. You have a low concentration outside the cell of potassium ion. So what happens is the cell will use ATP to move these low sodium inside the cell, a low sodium concentration, out to a high sodium concentration. That is the opposite of passive transport. That's active transport. And to do that, you need ATP. You need energy. So this little star is showing ATP breaking one of its 3 phosphates off. You can see the broken one off here, and releasing the energy to do that. And you see that same burst of energy causes potassium to move upstream against its concentration gradient as it moves potassium inside the cell from a low potassium to a high potassium concentration inside the cell. So this picture is again showing that diffusion, facilitated fusion, is fusion through a channel protein that will cause solute particles move from a high concentration to a low concentration. So this is a low blue particle concentration. This is the high blue particle concentration, whatever that blue particle is. So when you go from high to low, it does not require energy at all. In fact, it gives off energy that can be used to re-energize ADP into ATP, but we'll get to that later. So this is passive transport through a facilitated diffusion protein. Now, a protein that pushes from the low to the high, that direction, from the low solute concentration to a high solute concentration is pushing it up the concentration gradient or against the concentration gradient, and that requires energy. ATP is used to do that. That would be active transport, therefore, active transport AT, because it moves against the gradient as opposed along or down the gradient. Okay, we're going to go back to the cell membrane and talk about other kinds of materials in the cell membrane besides phospholipids and transport proteins. Okay, so we already talked about integral proteins. Integral proteins are embedded in both the polar heads and the fatty acid tails of the phospholipid bilayer. Now, some go all the way across, some go all the way across the membrane, such as transport membranes. So those integral proteins are called trans membranes. They go across. That's what trans means. Like transcontinental means across the continent. Okay, so that's one type of integral protein. Another type of integral protein may just be on one side. So it's not transmembrane. It's one means monotopic means side. You don't really have to know that. Just identify these guys if they're in the tails and the blue polar heads embedded there, then it's an integral protein. Peripheral proteins are just on the outside. Okay, so a peripheral protein can be connected to an integral protein, but they can be by themselves on the outside. Another lipid found inside the fatty acid tails is cholesterol. It's a steroid. And what's important for is keeping the cell membrane fluid so that these nonpolar tails don't get all glummed up to each other, because nonpolar things attract nonpolar things. They dissolve in each other. So it keeps it fluid, keeps them from getting all clumped up and rigid. You know, the cell membrane needs to be flexy, fluid. Okay, so you can see the cholesterol right there. We also have glycoproteins. Okay, glyco means, you know, sugar, like glycogen. So there's a carbohydrate chain, kind of like a polysaccharide, connected to a protein. So glycoprotein. Then we have also glycolipids, where you got a sugar chain connected to a phospholipid. So glyco means sugar, lipid. So glycoproteins and glycolipids are recognition molecules or signal molecules. And they're like little flags that tell the white blood cells that are on patrol looking for germs to kill. It signals to the white blood cell when it brushes by and taste actually those signal proteins and recognizes that, oh, this is not an enemy, leave them alone. If they don't recognize the glycolipids and glycoproteins on the cell, they will attack and kill that cell. So that's part of our immune system. Okay, so now that we described it, we can think of the model of the cell membrane, plasma membrane, as the fluid mosaic model. Okay, so the fluidy part is the phospholipids, and the mosaic parts are the proteins embedded or on the periphery of the cell membrane. So think of a mosaic as a picture. Like, I think of a stained glass window as little pieces of glass, and the grout that holds the pieces of glass is the fluid part. So for like I said in the previous slide, fluid part is the phospholipids, and mosaic part are like those little tiles. You think of a tile, mosaic as well, made of tiles. So the tiles would be the proteins. So proteins are like mosaic tiles in a fluid sea of lipids. Thus the fluid mosaic model. Okay. Now the peripheral proteins have several purposes. I'll just give you one. They're connected to, they call it peripheral proteins, surface proteins. They're connected to these protein fibers in the cytoplasm. And these protein fibers, where are they? They're these yellow strands. They're like what composes the inner skeleton of a cell. It's not hard like bone, but it's like a network, a tangle of protein strands that the organelles get connected to, and it gives shape to the cell. So the cell is just not a blob like an amoeba. So we've mentioned some functions of membrane proteins. We just mentioned how these proteins connect the membrane to the cytoskeleton inside that gives it inner structure, the cell. We went into a lot of detail about proteins that are transport proteins in the cell membrane. We talked about proteins that have little flags for recognizing, so that the immune system can recognize that this is friend, not foe, and leave him alone. But there's also receptor proteins. Receptor proteins are waiting there to attach to maybe a hormone, a signal from a gland that produces hormones, a signal that activates the cell to start some kind of chemical process to do something for the body that the cell's part of. So here's some more specific things. I already talked about what proteins do in the cell membrane. We already talked about that. The immune system recognizes those glycoproteins and glycolipids and leaves the cell alone instead of attacking it. Enzymatic. We haven't talked about enzymes yet, but these are receptor proteins that, again, like I said, when a signal hits it, it activates a chain of chemical reactions within the cell. And then cell-cell adhesion, well, yeah, cells stick together like skin cells stick together, or plant. A cell is not just free range when it's part of our body. It's connected to other cells. And that that cell sticking, those proteins stick to the proteins of other cells membranes, so that the cells can be tight together and work together in a tissue. Okay, so let's finish with a review. Which part describes the structure of a cell's membrane? Cell membrane. Protein sandwiched between two layers of, no, they're not, but they're not in between the phospholipids. They're either on the outside, or they penetrate from side to side. They're not stuck in the middle. Proteins embedded in two layers of phospholipids. Well, some are. Some proteins are like that. Those would be the transmembrane integral proteins. Phospholipids sandwiched between two layers of proteins. Nah, the proteins don't make two layers, and the phospholipids are in between. A layer of protein coating two layers of phospholipids. No, it's not like the proteins are two layers and the phospholipids like the jelly between the two pieces of bread. So no way. So it's letter B. Fluid mosaic model. Membrane structure refers to the fluidity of phospholipids and a pattern of protein. Okay, that's good. That's it. I'm not going to read on. Active transport. Moves molecules against their concentration using, yeah, that's exactly right. Energy is used in the solute particles are moved against their concentration gradients from low, again, that's from low concentration to high concentration, solute concentration. It's important to get that. Okay, what protein can be used for both active and passive transport? We did that way a while ago. So that'd be a carrier protein. He can let things through passively, and then he can change the course with energy and push them back out of the cell. So it's letter A. It's kind